Hello everyone. Today I will talk about the bioavailability of valine in a fermentation biomass product relative to the bioavailability in crystalline L-valine when fed to weaned pigs. Formulated low crude protein diets often makes it necessary to include crystalline amino acids in the diet, and by supplemented diets with crystalline amino acids, it is possible to maintain the growth performance in pigs fed diets with reduced crude protein. Valine is an indispensable amino acid which, along with leucine and isoleucine, belong to the group of branched chain amino acids, and the dietary branched chain amino acids are primarily used for synthesis of proteins. In weaned pigs, valine is usually the fifth limiting amino acid in corn and soybean meal diets. Therefore, in diets formulated by corn and soybean meal, if valine is deficient, there is a reduced efficiency of muscle deposition. And to maximize the deposition of muscle, dietary valine supplementation may be needed to meet the requirement. Feed-grade L-valine, which contains 98% of valine, is currently available for the feed industry and the primary source of supplemental valine included in commercial diets for pigs. Conventional crystalline L-valine is produced by harvest L-valine from the fermentation biomass. However, feed-grade L-valine may also be produced by spray drying the entire biomass, which will result in a final product containing 64.6% L-valine. It is expected that the bioavailability of valine in the fermentation biomass is similar to that in L-valine, but this hypothesis has not been reported. And therefore, we want to conduct this study to test the hypothesis that valine from a valine fermentation biomass has a bioavailability of 100% relative to L-valine when fed to weaned pigs. Moving on to material methods, this experiment used 224 weaned pigs allotted to the 7 diets and 8 replicate pens per diet, with 4 pigs per pen. Pigs were fed for 20 days. Individual pigs' weights were recorded at day 0 and at last day of experiment, and daily feed allotments were also recorded. Therefore, data for average daily gain, average daily feed intake, and gain-to-feed ratio for each diet were calculated. On the last day of the experiment, a blood sample from one pig in each pen was collected to analyze for blood urea nitrogen and the concentration of plasma-free amino acids. A total of 7 diets were formulated. Diets were based on corn, DDGS, corn gluten meal, and soybean meal. A valine-deficient basal diet was formulated, and 3 diets were formulated by adding 0.08, 0.17, and 0.25% of crystalline L-valine to provide a total valine in diets of 0.79, 0.88, and 0.98%. We had 3 additional diets that were formulated by adding 0.12, 0.25 and 0.37 of valine fermentation biomass to provide a total valine in diet of 0.8, 0.85, and 0.96% respectively. Moving on to the results, this graph shows the average daily gain for pigs fed diet supplemented either L-valine or valine fermentation biomass. The average daily gain increased as both sources were included in the diets. And moreover, pigs fed diet supplemented with valine fermentation biomass had a greater average daily gain than pigs fed diets supplemented with L-valine. The greater average daily gain in valine fermentation biomass may be a result of the presence of components other than valine in the biomass. The valine fermentation biomass also contains about 13.6% additional essential and non-essential amino acids. This graph shows the results for gain-to-feed duration. As a consequence of previous results, the gain-to-feed duration increased as the source of valine increased in the diets. However, we did not observe difference for gain-to-feed duration between crystalline L-valine and valine fermentation biomass. Pigs fed diet supplemented with either L-valine or valine fermentation biomass increased the final body weight compared with pigs fed the basal diet. Moreover, the final body weight was also greater for pigs fed diets supplemented with valine fermentation biomass compared with crystalline L-valine. The difference for body weight among of the supplementation level were 0.2, 0.6, and 0.8 kilograms more for pigs fed diets supplemented with valine fermentation biomass. This graph shows the results for blood urea nitrogen. The BUN concentration was reduced as either L-valine or valine fermentation biomass was included to the basal diet. However, no difference was observed in BUN between the two sources. The reduced concentration of BUN means that protein synthesis has increased in the pigs. This graph shows the plasma-free valine in pigs fed with L-valine and valine fermentation biomass. The plasma-free valine increased as the source of valine was included to the basal diet, and the increased valine concentration was expected and indicated that valine provided from both sources was efficiently absorbed from gastrointestinal tract. Based on this data, a linear regression was developed to determine relative bioavailability of valine in valine fermentation biomass using the crystalline L-valine as the standard. This graph shows the relative bioavailability for average daily gain, in which the equation shows that 126 is the common intersect, 409 is a slope of L-valine, and 597 is the slope of valine fermentation biomass. The slope duration was calculated by divided the slope of valine fermentation biomass and the slope of L-valine multiplied per 100%. With that calculation, we estimate that the relative bioavailability was 146% for valine fermentation biomass. The same way, the equation was developed for gain-to-feed duration. Based on this equation, it was possible to estimate the relative bioavailability for gain-to-feed duration in valine fermentation biomass was 135%. Based on that results represented to you, it was possible to conclude the relative bioavailability of at least 100% for valine fermentation biomass relative to L-valine. Pigs supplemented with valine fermentation biomass had a greater average daily gain and final body weight, and therefore valine fermentation biomass can be used as a source of valine in diets for William pigs. With that, I would like to acknowledge Evonik for your financial support in this study. Also, I would like to thank you for listening this talk, and if you are interested to know more about the research that we have been conducting, you can visit our website at nutrition.nsci.illinois.edu.